The overall goal of this protocol is to generate high-quality copies of the ordered, non-redundant transposon mutant library of Pseudomonas aeruginosa, strain PA14, referred to as the PA14NR Set.
This protocol preserves the integrity of the PA14NR Set during replication, generating high-quality copies of this library. The main advantage of this protocol is that it provides a detailed description of all steps involved in library replication, quality control, and proper storage of individual mutants.
Although this protocol was designed to replicate the PA14NR set, it can be adapted to replicate other bacterial libraries.
Generally, individuals new to this method will struggle because of the risk of contamination during the replication process. Therefore, this protocol provides detailed step-by-step instructions to reduce that risk.
Our laboratory distributes the PA14NR set to the global scientific community, and we recommend that the library is replicated by the end user. To provide guidance, we offer a protocol detailing how to generate high-quality copies of the library.
Visual demonstration of this protocol is critical, especially the steps designed to prevent cross-contamination.
For self-protection as well as protection of the bacterial cultures, a lab coat, gloves, and a mask should always be worn when handling the non-redundant Transposon Insertion Mutant library of P. aeruginosa strain PA14, designated PA14NR Set. Prepare LB agar plates by pouring approximately 60 milliliters of molten LB agar into rectangular plates. Dry the plates in a sterile hood for approximately 1 hour.